The overall goal of this in vitro procedure is to observe the effect of Zika virus infection on survival and differentiation of human fetal neural stem cells.
This method can help answer key questions in the neural stem cell and infectious disease fields, such as how viral infection influences innate immune responses and how these changes contribute to the survival and differentiation of neural stem cells. The main advantage of this technique is that the cells used are human and not genetically modified, which enhances the translational application of the results.
The implications of this technique extend toward therapy for the neurological deficits associated with the Zika virus because it provides a means to evaluate the mechanism of infection and screen therapeutics.
After preparing culture medium and plating human neural stem cells according to the text protocol, retrieve from the incubator the flask of cells to be passaged and bring it into the biosafety cabinet, or BSC. Gently tilt the flask, allowing the cells to sink to the bottom of the pooled medium. Transfer 10 milliliters of medium into two 5 milliliter portions into a clean 15 milliliter tube labeled CM for conditioned medium. Take care not to aspirate any of the cells settled in the flask. Transfer 3 milliliters of medium from the CM tube to a 15 milliliter tube labeled trypsin inhibitor solution, then set the CM tube aside. Next, remove the 5 milliliters of medium and cells remaining in the T75 flask and place it in a clean 15 milliliter tube labeled cells. Then, using a serological pipette,